Hey guys, so here we are in the benchmark review. Our benchmark is going to cover everything from the beginning of the year up through ATP, photosynthesis, and cell respiration. So everything that we've covered up to this point, not including the stuff on plants. All right, so the first thing that we need to talk about are the steps of the scientific method. And you've learned this like since middle school, so I'm not going to spend a lot of time. So basically, it starts with making observations and coming up with a question or a problem that you want to solve. From there, a scientist generally does research, so they've got background information about what they're trying to figure out, and they come up with some inferences, which are basically answers or what they think are actually occurring and why. From there, they use that background information and that knowledge to formulate a hypothesis. So in the past, you've learned that a hypothesis is an educated guess, and it's totally not at all. So I want you to get that out of your head. It's not an educated guess. It is backed by a lot of research and data and a lot of things that scientists do before they formulate that hypothesis. It's a statement. It's a statement about what you think is going to occur and what you think is happening, and it is going to be tested. So it's a testable statement. From there, you run a controlled experiment. In a controlled experiment, you change only one thing at a time. Those things that change are called variables. And in our experiment, we have several different variables. We have the independent variable, dependent variable, and then these things called constants. The independent is the one that the scientist changes on purpose. I like to remember independent starts with an I because I'm going to change it. I'm the scientist. I'm running the experiment. I'm going to change this variable on purpose. The dependent variable is your result, what you're going to measure at the end. And then the constants are things that stay the same throughout the experiment. So if we use this example, like I wanted to test that the fact that fertilizer makes plants grow the tallest. Okay, so I think if I fertilize a bunch of plants, they're going to grow really super tall. So my independent variable is going to be the amount of fertilizer that I give to my plants. The dependent variable is going to be what I measure at the end, the plant height. The constants are going to be things that stay the same, and basically everything's going to stay the same. So I'm going to plant the same kind of plants. I'm going to use the same kind of fertilizer. I'm just changing the different amounts of fertilizer. I'm going to plant them in the same type of soil, the same pots. I'm going to give them the same amount of light, the same amount of water. So the things that stay the same. In our experiment, we also have a thing called a control group. Our control group acts as kind of like a comparison for my experimental group. So my experimental group will be the plants that are getting all the different amounts of fertilizer. Then I'm going to have a plant that does not receive any fertilizer at all. That's going to act as my comparison, my reference. So that's my control group. It does not get what I'm testing. So it does not receive the independent variable. From there, I collect a bunch of data. My data can be qualitative or quantitative. Quantitative sounds like quantity, and it deals with numbers. So we use measurements like how tall, how much does it weigh, how many are there. So data collected by measurements using numbers is quantitative. Then we have qualitative data. Qualitative is data collected by using the 5 senses. So what does it sound like? What does it smell like? What does it taste like? What does it feel like? What does it look like? Okay, so data collected using the 5 senses. From there, we draw conclusions. We analyze that information and figure out what's happening. If we need to go back, we go back, we start over. Maybe that makes a new question when we get to the end. We haven't solved our problem yet. We need to reassess and start over, whatever we need to do. From there, if we've got a great experiment, we can move on to the peer review process where we submit our experiment to other scientists in the same field. That checks all of our information to make sure we're not biased and we're not like getting paid by a big company to pull this experiment in their favor. So it gets double checked and analyzed by other scientists before it gets published and put out for the public to actually learn from. So the next thing we talked about at the beginning of the year were scientific laws versus scientific theories. And I want you to remember this. Scientific theories are in-depth explanations. So they explain something that's happening. A scientific law is just a statement about something that's occurring. So it's like just basically a fact. It does not explain why. It just describes what's happening. Okay? So if you think about the scientific theory, the word theory has an E in it. If you can remember, a theory is an explanation because it's a detailed, in-depth explanation. Scientific law is just what is happening. Okay, we're going to move on now to water, and we learned about the properties of water. Well, water is a polar molecule. And why is it considered a polar molecule? Well, it's polar because it has an uneven distribution of electrons. If you look at my picture over there of the water molecule, you can see that the oxygen is super, super, super big, and my hydrogen is teeny tiny. So I've got two little hydrogens. And normally this is a covalently bonded molecule. But because the oxygen is so big, it doesn't share evenly. It's a big bully. So it pulls more of the electrons towards itself, making it more negatively charged, and then leaving those little hydrogens to be partially positively charged. Because of these little charges, this positive and negative end, it can form bonds to itself via these things called hydrogen bonds, which makes it do a lot of unique things. So there's several different properties of water. The first one being cohesion. Cohesion is where water molecules bond to other water molecules. We can also call this surface tension. A really common example of this is when one of those little water bugs, we call them water striders, walks across the top of the water, and they don't break the hydrogen bond, so they can just run across the top of the water real fast. So it's water bonded to other water molecules. Water forming a perfectly round little water drop is also cohesion, okay? Because water is bonding to itself, forming that perfect little drop. Then we have adhesion. Adhesion is water molecules bonded to other types of molecules. Because of this, we can call water a universal solvent. It dissolves a lot of different things. Because of its polarity, the positive and negative ends pull a lot of other molecules towards it and dissolve a lot of things. An example of adhesion is that little dip when you pour water in like a test tube or like a measuring cup, it forms that little dip in the middle. That's because the water molecules are more attracted to the sides of the glass than they are to each other, forming that perfect little dip in the middle. That's called the meniscus. Capillary action is actually how water moves up through the roots of plants all the way up to the top branches. It actually uses this stuff called xylem. Xylem is like a little straw inside the plant. It's in parts of the plant cells. And the plant cell actually moves the water through using both the forces of adhesion and cohesion. So adhesion, the water molecules are attracted to the sides of the straw, the xylem. And because one water molecule is attracted to other water molecules, when one moves up, the other ones move with it. So you can see that in that picture there. So capillary action is how water moves up through the roots of plants, okay, all the way up to the top branches. And it's mostly through the use of adhesion with a little tiny bit of cohesion. Our next two properties of water, we have high specific heat, which is also high heat capacity. And then we have less dense as a solid. But high heat capacity or high specific heat means that water resists changes in temperature. It takes a lot of energy to change the temperature. To even like increase or decrease the temperature of water by 1 degree would take a ton of energy. Okay, so this is why we don't have boiling oceans or frozen solid oceans, even in the Arctic, because it would take a ton of energy to change the temperature. And even in the winter around here, the Gulf only fluctuates a few degrees up and down, even though the air temperature might change a lot. It's because water has a high specific heat or high heat capacity. Less dense as a solid means that ice floats. So ice is less dense. Remember that, less dense than liquid water, so it floats to the top. Now, less dense means those particles are much more spaced out, like the picture I have out. When they freeze, water expands, okay? Like when you put a Coca-Cola bottle in the freezer and you see it pop up, that's because the water in it is expanding. It becomes less compacted, less dense. So it floats on the top of liquid water. We also talked about solutions in the pH scale when we talked about properties of water. A solution is a homogeneous mixture. Okay, so homogeneous just means it's the same concentration throughout. So just like our Coca-Cola bottle that I just gave you the example of, when you drink it, the first step all the way down to the bottom tastes the same. It's homogeneous. It's the same throughout. And in our solution, we have two parts. We have the solute, which are the little particles that need to get dissolved, and then the solvent, which is the liquid that does the dissolving. Then we also talked about the pH scale. And the pH scale goes from 0 all the way up to 14. Now at 7, that's where you would find pure water. That's absolutely neutral, so pure water. As you start down at the very bottom, so 0 all the way up to 6, those are our acids. And then from 8 to 14, those are our bases. So if you need a little trick to remember this, just remember that 7 is water, okay, in the middle, no problem. But A comes before B, so A's, the acids are the low numbers, and then the B's, the bases are the higher numbers. Okay, from there we talked about macromolecules. Macromolecules are large molecules, okay? So for our macromolecules, we have 4 categories. We have carbohydrates, lipids, nucleic acids, and proteins. Our first macromolecule, carbohydrates, are built out of small little monomers or building blocks called monosaccharides. Monosaccharides are simple sugars. You need to remember that. Now, carbohydrates, when you eat them, they break down into sugar in your body. So if you eat rice, cereal, pasta, they all turn into sugar in your body. So they serve as the main source of energy for our cells. This is why if you're an athlete, your coach might tell you, oh, make sure you eat a big bowl of spaghetti or something before a big game, because it's going to be a big burst of energy, okay? So carbohydrates are built out of monosaccharides. Those are simple sugars. They're used as a main source of energy for all of our cells. I forgot to say, when I'm talking about macromolecules, macromolecules are just those really, really large molecules. We call them polymers because they're made up of a bunch of little pieces together. That root word poly means many, because it's made of many, many pieces. Those little pieces are each called monomers. That's why I say the monomer that builds it, or the monomer or the building block, those are the monomers. And each of our macromolecules, or categories of macromolecules, each have a specialized name for their monomer. Okay, so remember, carbohydrates, monosaccharides, main source of energy for our cells. Examples are sugars and starches like rice, pasta, cereal. Also, this is the only one that's found in a 1 to 2 to 1 ratio. That means a 1 carbon to 2 hydrogens to 1 oxygen ratio. So if you ever get asked a question, it's like, this macromolecule is made of a 1 to 2 to 1 ratio. If they say anything about a ratio, it's a carbohydrate. Next one, lipids. Lipids are fats. So if you can remember that their monomer or building block is called a fatty acid, because when you hear the word lipid, it's a fat, you can match those up. So lipids are built out of fatty acids. Their whole job, their whole function is to store energy. So, you know, when you store a couple extra pounds, that extra fat that you're storing could be used for energy later on if you don't get enough food. They also surround our cell, they protect our cells, protect our skin. When we secrete oil, our earwax is a lipid, so it protects the inside of your ear. So some examples are fats, oils, waxes, and then also cholesterol. Then we have these things called nucleic acids. Nucleic acids are built out of building blocks called nucleotides. The nucleotides have 3 parts. They have a phosphate, a sugar, and a nitrogen-containing base. You will be asked that on your midterm, benchmark exam, and EOC. So please remember, nucleic acids are built out of nucleotides, and each nucleotide has 3 parts: phosphate, the sugar, and the nitrogen base. The whole job of nucleic acids, the function, is to store and transmit genetic information. Our examples are DNA and RNA. And in DNA, the Na stands for nucleic acid. In RNA, the Na stands for nucleic acid. This is probably the easiest one to match their examples and the actual macromolecule to its building block, because they both start with N. So nucleic acid, nucleotide, and then Na for DNA and RNA. My last one is probably the most important one for you to remember. So we're going to talk about proteins, and proteins are built out of little monomers or building blocks called amino acids. Now, even every year, they will ask you multiple questions about that same thing. So proteins and amino acids, or amino acids and proteins. Remember, they go hand in hand. They serve a ton of roles. They do many different things for our body. They control our traits. So they determine, if your hair is curly, straight, the color of your eyes, a ton of different things about you. They control reactions in our cells, they fight disease, and even transport substances around our body. An example of proteins are enzymes, and we'll talk about that here. So enzymes, what are they? Well, they're biological catalysts. And what do? All they do is speed up chemical reactions. That's their job. And they're very, very specialized. You have enzymes that do a ton of different things, but they all are specific. They only do one job. one enzyme for one job. Now, because they're so specific, if they are in the wrong place, they will not work anymore. And what I mean by that is if you have an enzyme that is in your stomach and it helps you break down food, if it then gets that, does its digestion process and it moves from the stomach into the intestines, it will become inactive because it will be in the wrong pH. So if it moves somewhere it doesn't belong, it becomes inactive. Same thing with temperature. If the temperature is wrong for the enzyme, it becomes inactive. It will not work. Now, what we need to look at here is this graph. Okay, this is showing the activation energy of a chemical reaction. Remember, chemical reactions require the input of energy in order for them to move, to react. So if we look at that red line is representing how much energy it takes to start the reaction. And this is without the use of the enzyme. It's just a normal reaction. If you look at that big hill in red, it's a lot of energy that it takes. Then we have a second line, the blue line. The blue line is representing the same chemical reaction, but with the enzyme added to it. And you can see the hill is much, much, much shorter. Okay, this is important because you will probably be asked to pick out this graph in a series of like 4 graphs showing the reaction without the enzyme and then the reaction with the addition of the enzyme. So remember, with the enzymes, the hill is going to be shorter, much smaller, and then obviously the one that does not use the enzyme will be much bigger. Okay, from there we're going to talk about microscopes. And you know what? I'm going to put this in a section 2 because I want you to answer some review questions about this first part. So in our next video, we will start here. All right, guys, I will see you soon.